Is cancer a recessive trait? This question is often asked due to the existence of tumor suppressor genes. Tumor suppressor genes are genes that, when expressed, are able to stop cells from becoming cancerous. So how is a cell able to become cancerous? Cancer cells have found mechanisms to block tumor suppressor genes. How is this done? Sometimes through genetic mutation of the tumor suppressor genes, either through inheritance of a mutated gene or exposure to a toxin that mutates the gene. Another way to block tumor suppressor genes is through mistakes made in coding for the genes during cell replication, or to silence the genes so that the genes can never be expressed. Considering cancer results after these genes have been blocked, the idea has been proposed that cancer is recessive. Thus, the dominance of tumor suppressor genes protects most cells from becoming cancerous. But what do these tumor suppressor genes code for? One key example is cell cycle arrest. Replication is essential for tissues, as it is when cells grow new copies of themselves to the point of repairing damaged tissue. However, replication should stop once the tissue is healthy again. The cell cycle is the cycle a cell enters that leads to replication. Cell cycle arrest is regulated largely by blocking the signals for the cell cycle to continue. pRB is a protein that arrests the cell cycle by binding to E2Fs. E2Fs turn on the genes of the cell cycle, but pRB can bind to E2Fs, thus blocking the cell cycle from being turned on. If pRB cannot bind to E2Fs, it would mean that E2Fs would be free to continually signal for the cell cycle to keep going and for the cells to replicate possibly without end. Thus, the gene that codes for pRB is a tumor suppressor gene, as it codes for a protein that can arrest the cell cycle and stop cells from replicating. Another crucial way to prevent cells from becoming cancerous is through apoptosis, which is the programmed death of a cell. When a cell's DNA becomes damaged or other signals arise, it is possible for cells to turn on the pathway for apoptosis. This means that cells that have damaged DNA often follow a program to die so that they will not enter the cell cycle and replicate with damaged DNA. Otherwise, the new cells would contain the damaged DNA. p53 is a key protein that allows cells to initiate apoptosis. Specifically, p53 can bind to genes that code for apoptotic proteins and initiate apoptosis when it is needed. Blocking the function of p53 would block apoptosis and allow a cell with damaged DNA to continue into the cell cycle and replicate when the p53 protein would have otherwise stopped the cell from replicating and could have even caused apoptosis of the cell. Thus, cells can become cancerous by living longer than they were supposed to, and these cells that should have died are instead making numerous copies of themselves through unregulated replication in the cell cycle. Tumor suppressor genes, such as the genes that code for pRB and p53, are able to stop the unregulated replication.